accurate are automated gap filling methods, which are frequently used in the reconstruction process of genome scale networks.
Thanks to the organizers for inviting me. Is this turned on? Turned on here. Can you hear me?
No.
Right. They can't hear me though. Is the mic working?
I think the mic is like it work for me.
Yeah, I think I'd rather use this if this is working.
It's working. Now I'm recording.
Okay, great. Okay, overview of my talk. I'm going to first describe what's the problem I'm tackling, namely evaluating the accuracy of reaction gap filling. Well, what is reaction gap filling? I'll then give an overview of the software tools that we use in my group for producing metabolic models and for gap filling metabolic models, which is the MetaFlux component of the Pathway Tools software. And then I'll talk about two different approaches to measuring the accuracy of gap filling. The first is to gap fill a model that has had reactions randomly deleted from it to see how accurately the gap filler will restore deleted reactions. And the second is to compare a manually gap-filled model with a computationally gap-filled model. Okay, so we've heard already two talks about flux-balance analysis. It's usually a steady-state technique that uses a set of constraints to construct quantitative models of metabolism. And for example, my group published a couple years ago an E. coli metabolic model using flux-balance analysis. Now, flux-balance analysis models have four basic components to them. They have a set of nutrients that are the input chemicals that the cells grow on. There's a metabolic reaction list, the reaction network that processes those input nutrients to produce a set of biomass metabolites, namely the biomass components of the cell, like the amino acids, nucleoside triphosphates, cell wall components. And usually there are some secreted waste products as well. And so when you're assembling a metabolic model, you need to assemble all four of those components. And what a quantitative flux balance analysis model computes, the outputs of running the model, are assignments of steady state fluxes to every reaction in the network, as well as a computed overall cell growth rate. So what's the gap filling problem? Well, the input to the gap filling problem is, first of all, a metabolic model that's not solvable due to missing reactions. Not solvable means the model doesn't run at all, usually because there are missing reactions. The reasons reactions are missing typically is because of incompleteness in genome annotations. So these genome scale metabolic flux models are usually assembled by starting from a sequenced genome, inferring what reactions are catalyzed by the enzymes in the genome, and using that as your reaction set. Well, because all genomes are incomplete in their annotations, the list of reactions is almost always incomplete, and therefore often models don't run. And the nature of how models are run is through solving a linear program consisting of a set of constraints assembled from the reaction set. And the nature of these models is that if there's even one metabolite that can't be produced through the network because of a missing reaction, then there will be no solution at all to the model. The third and final input to the gap filling problem is an external reaction database that the gap filler will draw reactions from. So what the gap filler software tries to produce when it's solving a gap filling problem for a metabolic network, a metabolic model, is it tries to propose a minimal set of reactions, R, from this external database, such that if these reactions are added to the model, then the model becomes solvable. So the gap filler is trying to tell you which reactions are missing from your model, and the assumption is that your external database contains all of the missing reactions. It's not always true, unfortunately, but that's the assumption. And there's been a good bit of past work on gap filling. A number of past gap fillers used mixed integer linear programming to formulate the gap filling problem. There's a piece of software called GapFill that uses either KEGG or MetaCyc as its external database. Model Seed used to use an MILP gap filler. They recently switched to a linear programming-based gap filler using a technique that we published in my group called FastDev. There's another linear program-based gap filler called FastGapFill, although it doesn't guarantee a minimum reaction set. And then there's another piece of software called Mirage that uses randomized reaction selection and a greedy heuristic to reduce that randomized set, and also uses some external information like phylogenetic and expression profiles. Okay, so we use a Flux-Balance modeling tool that we developed in our group called MetaFlux, and MetaFlux is a component of the overall Pathway Tools software system. So Pathway Tools is a very comprehensive genome informatics and systems biology software environment. It's actually one of the largest software systems in bioinformatics as far as I'm aware. It's been licensed by more than 9,000 groups to date. It's the equivalent of about 1.5 million lines of C code, although it's written mostly in Lisp and JavaScript. And it has a number of different capabilities. It lets you create and maintain an organism-specific database that integrates genome information with pathway information and regulatory information. So it's the software environment that we use to develop the EcoCyc database and the other members of the BioCyc database collection. So it has many different search and visualization and editing tools. The editing tools are what our curators use to update these databases. It has a whole set of genome informatics tools, from a genome browser to sequence pattern search, that's implemented using an external tool, PatMatch. It also has a number of pathway informatics capabilities. In my other talk at the conference, I'll be talking about a multi-organism, microbiome-oriented metabolic root search capability that we've added recently to the software. It can also perform qualitative metabolic reconstructions. It has a regulatory informatics toolkit, as well as a number of different omics data analysis tools, the newest one of which is called the Omics Dashboard, which we published about a year ago. And finally, it can also generate from the pathway genome databases that the software creates flux-balance analysis models for both individual organisms and for organism communities. So here's the overall process that you take using pathway tools to create a metabolic model. The input to the process is an annotated genome, and the pathologic component of the software infers the metabolic network of the organism by inferring the reaction set of the organism from the sequenced genome, by considering both the EC numbers and the enzyme names and the gene ontology terms that were assigned during the annotation process. The software also infers metabolic pathways by scoring each pathway in the external MetaCyc database with the score that indicates the likelihood that that pathway is present in the organism based on the enzymes from the pathway that are present. So the resulting pathway genome database that comes out of that process is where MetaFlux draws the reaction list from when it's formulating a metabolic model. The user, in addition, provides an external file that defines the nutrient set, the secreted metabolite set, and the biomass metabolite set for the metabolic model. And then the MetaFlux component of Pathway Tools will use a multiple gap filler technique to propose not only missing reactions, but it can also, in fact, propose missing nutrients and missing secreted compounds, although in this study we're really just focusing on the missing reactions component of the gap filling. So a bit of information about MetaFlux. It actually has 3 modes of operation. It has what we call solving mode, which is where you run a model, which you can apply to individual organisms and to communities of interacting organisms. You can run it both in a steady state mode and in a dynamic mode. You can run it over a single compartment or a 2-dimensional spatial grid with diffusion implemented. It also has a knockout mode, which is essentially running the same model over and over again with different genes or reactions knocked out. And finally, it has a model development mode, which is where the gap filler tool exists, and it will not only suggest reactions to add, but it will identify, even if you add as many reactions as possible, it's still possible to have some biomass metabolites that can't be produced. The tool will identify which ones those are, and it will also identify which are the blocked reactions and the blocked metabolites that may be preventing production of some biomass metabolites. Okay, let's skip that one. Now for our external reaction database, when we're doing gap filling, the database that we use is the MetaCyc database developed by my group. And I just want to point out that MetaCyc currently has about 40% more reactions than KEGG does. And so basically the larger a reaction database, the more likely you're going to be able to gap fill successfully because you basically have a larger background set of reactions to draw from. Okay, so how do we do our first assessment of gap filling accuracy? Well, we used the EcoPsych metabolic model, and we were modeling aerobic growth on glucose involving 87 biomass metabolites with 476 of the E. coli reactions carrying non-zero flux. So in any given growth state, most of the reactions are actually not active. For example, all the reactions that involve catabolizing other carbon sources besides glucose, they're all turned off. So what we did was to randomly remove reactions from our model. So if R is the starting set of reactions that are in the model, we transformed R to R prime by removing a certain set of reactions from the model. And we removed either one reaction at a time, or two reactions at a time, or 5 or 10 or 20 or 50 reactions at a time from the model, selecting at random. And we ran 100 trials of each of those numbers of removed reactions. So there are 6 different sets of reaction removal, and we did 100 trials, so that's 600 experiments right there. We actually did a total of 7,800 experiments involving some other combinations. So there were 13 total different combinations that I'm going to explain here. First of all, we looked at 4 different gap filling algorithms. And I'm going to refer you to this publication at the bottom for the details, but the basic idea is that one of them was linear programming based, the others were mixed integer linear programming, and techniques A, B, and C were all somewhat different ways of formulating the gap filling problem as MILP problems. Okay. And we were interested in the relative trade-offs between those formulations. Now, in addition, we tried 2 different solvers for solving the MILP problems. One is the commercial Cplex software from IBM, and the other is a free piece of software from a group in Germany called Skip. And for the LP problems, we only used Skip. In addition, we used 2 different mathematical ways of formulating the constraints for MILP, one of which is called Big M and the other is called indicators. That's also described in more detail in the paper. And so 3 gap filling techniques times 2 solvers times 2 MILP formulations is 12 combinations, plus the LP formulation is 1 more, so 13 times 600 gives our 7,800 experiments that we did. And I'll summarize the overall results in just a minute, but a few observations about the results we got. First of all, sometimes, okay, so we removed a certain set of reactions, we'd run the gap filler, and then we compare the reactions the gap filler proposed against the reactions that were removed. And if the gap filler was doing a perfect job, it would propose the exact same set of reactions that we had removed. Okay, well, sometimes we actually got a null solution. That is, sometimes the gap filler said, my solution is the empty set. Well, that's actually possible because it may be that when we randomly remove these reactions, we remove some non-essential reactions. because the E. coli model actually has, quote, redundant reactions. Not every reaction is essential. Not every reaction, if removed, will prevent growth of the cell. So this shouldn't be too surprising. In addition, for a similar reason, sometimes the solutions actually contain fewer reactions than were removed. So this would be the case if some of the reactions that we removed, let's say we removed 5 reactions and one of them was non-essential, well, you could gap fill with 4 reactions to restore growth. Now, sometimes the solutions contain more reactions than were removed. And this is a surprising case. This was a big surprise to us during the study because the gap filler should have been able to, every reaction in EcoCyc that was being removed was present in MetaCyc. Okay, so the gap filler should have been able to find a solution that was at least as small as the set of reactions we removed. And further study of this indicated that it was actually numerical errors by the solver that were causing the solver to return non-optimal solutions, solutions that were larger than they needed to be. Another observation was that when as few as 10 reactions were removed, hardly any of the solutions exactly matched the reaction, the removed set of reactions. And this is because the number of solutions is increasing combinatorially, and the more reactions that are being gap filled, the smaller the probability that the solution found exactly matches the set of reactions removed. And so in the following, I'm going to report precision as the number of reactions removed that were suggested by the gap filler in its solution divided by the number of reactions suggested. So what fraction of the suggested reactions were in the original removed set. And I'll specify recall as the number of reactions removed that were suggested by the gap filler divided by the number of reactions initially removed. Okay, so here are the overall results. So these 2 rows are precision. These 2 rows are recall. And along the top, we're seeing technique A, technique B, technique C, and then the LP approach. We're seeing big M versus indicators, big M indicators, big M indicators for each of the techniques. And then we're seeing skip and Cplex results for each technique. So let's start with one interesting thing we found, which is that when we ran skip with the indicators method, it always timed out. It was way too slow. So that's one thing we learned is you don't want to use indicators with the skip solver. It's way too slow. Overall, Cplex gave better results than skip was another thing we observed. I think it's the case that, so the bold numbers are the best results for each of the methods, and it's always the case that the bold numbers are in the Cplex row. So Cplex is a commercial solver, and it does do better, but it's a bit expensive. Let's see. So to summarize these results, what we found was that overall, at least 71% of the reactions that were suggested were correct, were originally removed, but that means 29% of the reactions suggested were incorrect. And also, more than half of the correct reactions were recovered by the 4 gap fillers. It was commonly the case that solutions were smaller than the true set of reactions removed, again, because the reactions that were removed often contained non-essential reactions. And so one overall observation is the cell doesn't always use a minimum set of reactions. So kind of interesting. We kind of like the Occam's razor principle, but the cell is not always using a minimal set of reactions. Okay, so to summarize the differences between the 3 techniques, for technique A, 24% of the solutions didn't grow. Okay, so this is another big surprise, is that we actually checked all the solutions by running all the gap-filled solutions as models to see, did the models actually grow? Well, for technique A, 24% of the time they didn't grow, which means the solutions were wrong. So the technique A formulation produced a lot of bad solutions, which was not the case for technique B or technique C. Another important distinction between technique C and B is that C could identify non-producible biomass metabolites. The technique C formulation could tell us which of the biomass metabolites were not being produced, which is important debugging information to know, whereas technique B could not do that. Okay. We're running out of time here, so let me now take you quickly to approach 2. And this is, approach 2 was basically to compare a computationally gap-filled model with a manually gap-filled model. And we used this organism, and what we did was to start with its genome sequence, annotate it using K-base, run that annotation through pathologic to create a qualitative metabolic reconstruction, and then subject that same starting database, which had gaps in it, to both manual gap filling and automated gap filling. And I'll wrap up quickly here. And in its initial gap form, only 15 out of 53 biomass metabolites could be produced. So, but it turns out that the computational gap filler proposed adding 12 new reactions to fill those gaps. The manual solution involved adding 13 reactions, of which 8 of them were shared with the automated solution. And so comparing predicted result to manual solution, we get a recall of 61% and a precision of 66%. And I'll just wrap up by saying we do need more studies like this. One study of one model using one gap filler is not all that definitive, so I encourage those of you building models to do similar studies. It is tricky to do this unless they share the same reaction database. So if the gap fillers are drawing from different reaction databases, that's going to modify that's going to influence their accuracy a lot and interfere with a strict apples-to-apples comparison. So here are the overall comparison results. In both approach 1 and approach 2, we got very similar recalls. The precisions were different. Here I chose the technique that we are going to end up using going forward in MetaFlux for a variety of reasons. So thanks for your attention, and happy to take questions if there's any time left.
Unfortunately, we have to postpone all questions to the coffee break. So we thank Dr. Karp for his talk. And now I announce the next speaker, Zahid